So we're going to move away from air pollution and speak more broadly about environmental disruption and infectious diseases. And Justin Remais from the University of California, Berkeley will talk to us about climate change.
Thank you, Josh. So privileged to join you today and to be in a room not only where there are those who focus on chemical stressors and those who attend to infectious or biotic stressors, but also from molecular to global. It's really been an incredible day. I'm an engineer by training. My research group at Berkeley focuses on, well, some of those larger scale issues, and I'll try to reflect a bit, drawing on some of my work on kind of where we started the discussion today with Robert's, Laura's, and other talks, and where I see environmental health science contributing to an understanding of infectious disease, kind of working up that scale. I would say, too, we work on West Nile virus and coccidioidomycosis in California. We have a major emphasis of work in West Africa on helminth infections influenced by pesticide exposures in interesting ways that I'll get to. And we also are invested heavily in research in China with collaborators. Here is a cityscape of Shanghai, and many of your eyes, given your training in this room, are drawn towards it as an affluent setting, a gleaming city by this depiction, but also a center for major air pollution exposure, heat islands, and heat stress. Those of us with an infectious disease orientation are thinking of diseases transmitted in very high-density urban settings, including TB in Shanghai, water systems affected by Legionella or Cryptosporidium, vector-borne infections that are urban transmitted, including dengue in Shanghai. So Shanghai is somewhat of a microcosm of many of the ideas and endpoints and risks that we've discussed today. And I'd like to make an argument initially that to go back to some of the large, more distal drivers of health that we heard at the outset, urge a greater involvement in their characterization from the health science community. That is, being the ones who are not only seeing these as this defining domestic and global kind of context in which public health and environmental health is being practiced, but also a need for us as health scientists to be involved in their characterization. And so here, I'm going to suggest that we capitalize on some of our expertise methods to better understand, yes, there are infectious disease consequences, but also them as socio-environmental phenomena themselves. Doing so, in my view, has a few benefits. We'll have a characterization of the phenomena that's more in line, perhaps, with the health analysis that we want to conduct. Doing so will ensure that our colleagues in the environmental sciences and engineering sciences are having that characterization infused with the health perspective. And also, and here in a series I'll share with you now, it gives us a chance to map out the experience of major global environmental change in the health literature. Here are some examples of that. Work that I led or contributed to looking at rapid and unplanned urbanization in China, the largest human migration in history that is occurring over the course of decades, what took more than a century in our own and the Western experience, and understanding how exposures are shifting commensurate with China's urbanization experience and that which is playing out around the world is our task. But understanding that urbanization phenomena, and here is one example of that, is as important. Here we established essentially the definitive kind of interprovincial migration pathways that played out over the 3 decades into the current century, from somewhat modest green arrows or vectors between pathways to the very intense movements of people, especially into the formerly largely rural Western provinces in China. This is, yes, influencing exposures to urban air pollution from formerly rural populations. It's changing diets, many consequences. For infectious diseases, it's bringing people into contact with an inadequate protective infrastructure like water and sanitation. It's bringing immunologically naive populations into contact with pathogens that they hadn't had prior exposure to. It's straining health systems and doing much more. we carried this work forward to understanding transport in public health, and many of you in the room will think air pollution, impacts of transport on the global climate system, accidents and injuries. We add to that the unprecedented mobility that China's motorization, the development of shared transportation means, including the shared bike transport that China pioneered and that now we're seeing in American cities, including this one, the largest high-speed rail network in the world, and what this means for the spread of infections that are either directly transmitted or transmitted through environmental media. A couple other examples of the work I think we can do here, thinking about scarcity and mapping out resource scarcity as a public health issue, and being sure that those ideas are represented in the health science, especially the health literature that is physician facing. And so Lancet has been a great partner for many of us in this effort, including through the Planetary Health Initiative that they were so central to. So water scarcity in China that we have thought quite a bit about here, the density of China's population in the east, where coincidentally and as a major driver of where water resources are most strained in the dry northeastern areas, where the plus signs represent per capita water resources, and the most constrained resources are, of course, where the people are. Those are also the driest areas. China is working to address that with a set of challenging engineering projects, much like California did in its aqueduct system. You would need a water transfer project many times that of California's to accomplish sufficient south to north water transfers in China. If there's one thing that China is good at, it's large-scale engineering, and so that work is underway. Health consequences of that, especially for waterborne diseases, are something we're interested in. Food safety and food supply is another major focus of those of us who are interested in global change. And in China, the experience with water shortages, the experience of urbanization, claiming formerly agricultural land is pushing down the arable land per person in China and putting people and human settlements much more proximal to agricultural activities that have consequences for pesticide exposures and others. This, of course, is coming right alongside changes in diet that are increasingly based in animal proteins, and that has unique consequences that we've heard about this morning for waste management, for the transmission of pathogens that take advantage of high-density animal agriculture to find their way to make contact with human hosts, especially workers. I'll make one last comment in this series, and that is on climate change. Here is probably where we've been most integrated, kind of from the most distal characterization through to health analysis, and there are reasons for that I'd love to discuss. But here is an opportunity for us to perhaps do more in the health analysis, especially the forward-looking health analysis, in that projection space that we have some unique training to offer in. So let me just conclude here by saying we are counted on to understand these phenomena. And in fact, within the health sciences, if we don't characterize them as environmental health scientists, there probably aren't other health scientists who may be able to step in, especially those with the kind of quantitative tool set and the attention to methodologic detail that we might bring. Our infectious disease colleagues, in my experience, have made it clear to me that they're very interested in us doing so in order to kind of build the partnerships that they need in order to do more rigorous infectious disease epidemiology infused with an understanding of these important societal shifts. And fortunate, though, that we're ready to help and we're well trained to do so. These are members of my group with training to deal in mass balance and contaminant partitioning and fate and transport modeling, dealing with challenging exposure estimation issues, understanding complicated heterogeneity in dose response and in infectious dose, and so on. So we're ready for the challenge. If I had to lay out 3 areas where I think environmental health science has the most to contribute to global infectious disease research and converting that research into public health action, I would say measurement, modeling, and projection. If I had to point to kind of special skills of our area to really push the bounds of an understanding of infectious disease as it relates to to environmental influences, these are the kind of categories that come to my mind. And let me start with a couple examples of each and start with measurement. Detection of contaminants in environmental matrices, something that we all start with in the environmental health sciences when we're trying to understand a hazard and then the interactions with the environmental medium that might lead to an exposed population. Here is one detection challenge for an environmental pathogen. This is Schistosoma japonicum here, one of 3 schistosomes that cause schistosomiasis. More than 200 million people estimated infected by one of the 3 human schistosome species. A very important waterborne or water-related infection. And one question that we might bring first is, well, how do we go about measuring its presence or its concentration in the environment? It's 2019, I'll remind you, and this is the best current way we have for estimating the concentration of schistosoma species in the water column. Mice are dangled up to their bellies for a discrete exposure interval where they are potentially exposed to the pathogen while present in the water column. Then they're then taken back to the laboratory where they are raised for about 6 weeks while the parasite develops in vivo. Any parasites they encounter, they're then sacrificed, and the number of established worms are counted in their vasculature, and then that's used to back calculate the number of pathogens that they must have encountered in the waterway given the flow characteristics and the channel morphology in order to estimate concentration. Right? A hugely costly, very poor spatial temporal resolution, ethical issues associated with using animal assay or an animal model in this particular mode of surveillance. If you think this is limited to pathogens that are present mostly in low and middle income countries, or those that are part of that suite of neglected tropical diseases for which schistosomiasis is one, we are now working on coccidioidomycosis in California. It's a dust-transmitted fungal agent that blows in dust in arid areas. California is experiencing the highest recorded incidence ever. This is true also in Arizona in the last year. The most recent year, 2018, is set to top that number of cases. It's a major source of community-acquired pneumonia in California, especially a risk for workers, has major morbidity associated with it, including the need for the most severe cases to receive lifelong antifungal treatment that has other health effects associated or side effects associated with those treatments. And to skip to the end here, we know very little about where this pathogen is because we cannot detect it in soil. And it has only been maybe in the 50s, as the quote from recent work from a really fantastic group at Northern Arizona who's been working to solve this problem, that any isolation from air samples has been undertaken. So here we have a pathogen that is causing significant disease in a place like the southwestern U.S., for which basic detection methods have been lacking, hindering our understanding of environmental sources. We don't know much about how it disperses across the landscape, how close you have to be to a putative source. We know nothing about exposure and can do little then to protect workers, especially, let alone some of the other determinants of severe or disseminated disease and so on that would come from a quantitative understanding. So as my colleague Joe Eisenberg and I put it recently, our tools are just grossly inadequate to take environmental action with respect to many important diseases associated with environmental pathogens, including just the basic means to detect the pathogen in the environment. We've done some work led by Berkeley, former Berkeley students, YY Hung and Amanda Driscoll, to develop a means of taking a grab sample and determining and quantifying the number of cercarial pathogens. I tell this story first in that measurement category because it just reminds us that in our field, the tradition of primary measurement of field science and field research is one that isn't necessarily matched, especially with our focus on environmental media and the dynamics within the media of pathogens by our colleagues within part of the infectious disease space. So it's something to draw upon that strength as we are looking for how we can influence the field going forward. A couple thoughts in the modeling space where our kind of special powers may reside there, if I had to describe some. What do we do? The first example, when what might we be able to contribute when we don't know much about the environmental hazard? And I'll use an example here of all that we know about extreme weather, especially extreme precipitation, and the influence on gastrointestinal illness. We know that from a large literature that extreme weather, especially extreme precipitation, has consequences for diarrheal disease. Major unresolved questions surrounding how much we actually know, given our ability to capture an exposure as fundamental as extreme precipitation. You'd think we could just, if it's something to measure where you can just lay out a gauge, you would think we have a good handle on it. Here, work led by postdoc Morgan Levy, who's now at UCSD, she asked, what are the pitfalls in estimating even fundamental exposures that cause an important burden of global disease through, in this case, the both endemic and epidemic gastrointestinal illness that accompanies extreme precipitation for those who rely on surface water, drinking water sources. What are the consequences for our epidemiologic understanding? And two approaches she took here. Since time is short, I'll just say that through both a simulation analysis and an analysis of a cohort in northern Ecuador that collaborators at University of Michigan have led, evaluated the bias in our estimate of that extreme weather diarrheal disease association. And that last plot, it's always hard to go backwards, is rainfall as viewed from different data sources, including remote sense sourcing, as well as different rain gauges in a Gulf Coast environment and a Pacific Northwest domestic environment. Basically, rainfall is a very patchy process, and our understanding of it is very dependent on the information we draw upon. What are the consequences for our epidemiologic understanding of diarrheal disease rainfall relationship? Well, it's that the limited fidelity that's typical of these rainfall products, if you will, these rainfall scenarios are sufficient to bias downward that estimated health effect between heavy precipitation and diarrheal disease. And importantly, that from simulation work, when we analyzed a cohort in northern Ecuador where 3 years of weekly follow-up on diarrheal disease, similar biases were evident, especially where there's spatial incompatibility between where we're measuring rainfall and where we're measuring disease. So the summary here is, yes, we can advance approaches to measurement, and that's a major commitment of many of us, but we also are in a unique position bringing our critical eye, our treatment of uncertainty, our accommodation of a rigorous accommodation of bias, time-varying confounding, and other aspects of environmental epidemiology that we pride ourselves upon to understand where the gaps in understanding in the infectious disease epidemiology realm. This can have very practical consequences, including being able to look at those prior estimates and understand, in this case, that they are likely to substantially underestimate the impact of extreme weather on infectious outcomes, which is what Morgan's work concluded, and why there was a great deal of interest as it came out. Two other areas of modeling, I would say. One is a kind of integrated model that would include not just environmental forcing, but also the interaction between social and environmental forces. So taking that same example of extreme or climatological determinants of diarrheal disease, what if we ask what the social dynamics that may coincide with those environmental dynamics, what the consequences of those are for infections? And I'll skip to the punchline on this. In the same study region, we asked, well, what about the social gatherings, the mass gatherings that we know are associated with infectious diseases because they put pressure on water and sanitation resources, they increase contact rates, they involve oftentimes food sharing, it can limit access to hand hygiene, they can involve disease that's imported by visitors. So we know that gatherings are important. Well, what happens when we use the set of environmental epidemiology tools to ask what the consequences of these social gatherings are in the context of disease that's largely driven by other environmental factors, including rainfall? The answer is substantial. Here we use distributed lag nonlinear models in this GEE framework, one, by the way, that was established and kind of pioneered for understanding the heat mortality relationship and in air pollution epidemiology. And here we are translating those methods for application to analysis of a cohort following a set of communities with respect to their diarrheal disease experience, and establishing just how important these moments where the communities come together are for driving diarrheal disease in these settings, especially the effect size quite larger on the right side, those community gatherings that have lots of travel, lots of crowding. Why is this important? Because we may not want to intervene to stop community gatherings and limit, therefore, all the things that make life wonderful, but we might want to act with respect to environmental intervention right alongside these community events. They represent an opportunity to engage in environmental health prevention, even as we would like to sustain those social activities in this setting and in all settings. So that was work by Phil Collender, bridging social dynamics and environmental dynamics, and funded by a cross-cutting NSF project that is coming to its conclusion at the end of this year. The last example in the modeling space I'll give, and I'll just say a word about it, is this integrated assessment tradition that we have in the environmental health sciences and how we can apply it to infectious endpoints. Here, we're studying through a joint NSF-NIH EEID project a relationship between helminth dynamics, helminth infections, so infections with worms, and the use of fertilizers, herbicides, and insecticides, as they have a set of kind of cascading effects through the ecosystem in which defines the ecology of this parasite. I won't talk about all of those pathways, although I would love to do so afterwards. But I will say that stitching together our understanding of the dose response for the ecological response, the ecotoxicological response across the full pathway, the set of pathways, is a major challenge that environmental health science is up for, in that this is what we do. We bring together diverse information on dose response, and we're prepared to kind of tackle the challenge of integrating that understanding in a coherent analysis, a meta-analysis here where we look at different agrochemicals and their influence on different aspects of the life cycle of schistosomes, and then importing that information into integrated here, a dynamical mathematical model that integrates in the colored parameters, if you will, the influence of agrochemical exposures on the different aspects of the life cycle of this parasite, here fit to a community in which this work is embedded in West Africa. What can we do then by chemical, by pathway, establish what the influence on R naught, that summary parameter we use to describe the kind of strength or the force of transmission within a population, and we can look across concentrations and in particular at the expected environmental concentration at the effects on the R naught of the infection in a particular community, noting that there are ways in which these same chemical exposures through different pathways can both have an adverse consequence, intensifying transmission, positive R naughts, as well as those that are protective, the blue and red paths that as atrazine increases through direct toxic effects on the larval stage of the parasite in water, for instance, atrazine can be a good thing for reducing schistosomiasis. That is sure to be a controversial statement that I've made that will forever last, especially with the web recording. And it turns out that these are component effects, and when you put them together, the nonlinearities in the system actually favor the green response for reasons I won't go into. So here, this is an opportunity to bring together the related fields, ecotoxicology, ecological theory, population biology especially, and epidemiologic methods in a unifying platform where we can also bring in economic analysis and make this kind of modeling enterprise look a lot like the integrated assessment models we use in other areas of environmental health science, like energy systems analysis, air pollution, and other consequences. Last area that I'll just mention without showing you any work is projection. We have a kind of special relationship with forward-looking analyses that is not common when, as I've thought about it, within the health sciences, right? We are willing to make projections 10, 20, 50, a century out in ways that are quite different from many of our colleagues in the other related areas of health science. And in part, that's because the drivers that we are interested in, dependent on themselves, can be projected forward. Our dependence on certain energy sources for electricity generation and the kind of scenario analyses we might do there, our investment in water infrastructure and what we might learn from future changes as water systems are rolled out. So the last category I would say is that we have a special opportunity to bring projection into the infectious disease community because of the dependence of these parasites on environmental systems themselves, that we have gained quite a bit of comfort projecting forward in time and under a set of policy options. We did that in part in this work in an integrated assessment model that takes not just climate-sensitive diseases and climate change into account, but also the other things that we can project in WASH infrastructure, economic development, migration, and demography. So last slide, then, I'll say, is, in summary, our chance here is to make contributions that are, yes, reach from focused measurement all the way up to these far-reaching synthetic analytical scales. And in environmental health sciences, we have quite a few kind of toolkits to offer the global ID community. And from my own experience, they have been hungry to collaborate. And our colleagues will look at us to better characterize and understand those upstream drivers that are really within our domain in environmental science, environmental engineering, and yes, environmental health, so that we can do better, especially in that forward-looking mode: planning, strategy, resource allocation, policy, and advocacy. I'll end there. Thank you.